I will now work over the IBDP Biology IA draft for anonymous student N. Looking at this, light exposure, what are we talking about? Is this light intensity, light level? It's a little bit unclear. Secondly, whereabouts are we talking? No idea looking at this. Thirdly, how is it measured? To make sure things are easy to read, add a gap between paragraphs. You're not constrained by a space limit, it's a word limit, so help your readers. The main variable is light. It's not well covered here. It should be here, as well as more on benthic invertebrates. Probably we just call that light levels. Good to see units. Caused by what? Probably you need to say something about caused by coverage of trees growing on the sides of the bank. Okay, this is table 1 and it has a title. We need a few more details on it as well. Good title. We need numbers. Measure and kept similar. Would you give yourself a mark for that? Waffle. At what depth? You need to add numbers for this column. Looking at your method. Okay, use Luxmeter on what phone? We need more details. 3 trials in each spot. What spot? Is this the center of a quadrat, center of randomized coordinates? Maybe this step needs to be moved to after you've actually done something about identifying your randomized coordinates. The whole thing here, my understanding is you had 2 large sample sites and then you randomized within those. And I don't see that information up here where you're already measuring things before you've defined those spots. This is really here. I think, there was some sort of randomization to get to the trials, to get to the spots you were using. A little bit more here on where in each site, what depth. Now you have 5 sites for each area. Where is this? It's not particularly clear. Compare and use? No. Calculate. For what? For each site? So you've got an average and a deviation for the pooled sites for light and less light? It's not mentioned here. Then you compared it with a t-test. Maybe you want to write that here too. Looking at this whole method, I think you need more sequencing to have it in order. Something about find an area. What made you choose that area? Well, this is very vague. How did you choose your area? Then within each area, your randomization. Then what you did within the area. And finally, what you did with the data after. So try and get it in order. This really should not be split over 2 pages. Control Enter or Command Enter on a Mac, and it's all together. Looks like we've got a problem here. Hard to read. Fix it. And the font size. Shrink it down to fit. I mean, really, just ouch there. Good to see consistent decimal places. Oh, except here. Why would one number only have 1 decimal place? Good to see average and standard deviations have that. However, how was this calculated? My guess is you used a formula in either a Google Sheets or an Excel. Make sure you say this before you get to SDI. Now, my next concern, this is not the version that we use for DP biology. So there's a problem. Now, just to get really hardcore in statistics, I suggest that you say because the variance was unequal, say what? Basically, your standard deviations are different. You couldn't use a standard t-test, so you used the Welch's t-test, which again is on the Social Sciences website, which I recognize this from. Looking at your graph, diversity, what are we talking about here? Is the Simpsons Diversity Index? Good to see the standard deviation labeled. Perhaps a lighter bar color to see the underneath part of the deviation, but all good here, mostly good. If you are going to make a claim here, not supported, you need to be telling us t-test, p-value equals da-da-da-da. Give us the details or the evidence for your statement. You need to talk about standard deviation, but talk about the overlap in bars. Does it suggest there's a high overlap and there's not much difference? Does it suggest a low overlap? The size, they're quite large for the difference between them. Things to comment on. Looking up here, standard 117133, the second one should be fractionally larger. I think it is, that's good. Now, talking about your conclusion, your first strand is all about what you found out. your conclusion, your results, your statistics, your discussion of error bars. The second strand is about comparing to published work. If you don't have that, the maximum you can get is 3 out of 6. I don't see that section here, so you really need to focus on adding this in, comparing to other published work, similar studies, similar rivers, what you can find. It might agree with what you found, it might disagree, but that gives you something to discuss to get the second half of your marks for your conclusion. Looking at your evaluation, I have 3 suggestions. The first one, a table with your limitation in the first column, the importance of the limitation, finally, the solution for next time. You need all of these. The first 2 will give you the first strand of evaluation. This will give you the second strand of evaluation. The second thing is to look at each of your cells. If you are generally stating the problem was this, you are aiming at a 1-2. If your limitation and your solutions are described overall, you're in the park for a 3 or a 4 for this criteria. If you actually explain the problem and then explain the solution, you're in the 5-6 basket. The final thing you need to do to get your 5-6 is to talk about the relative importance of each of your identified limitations or problems. Once these things are done, you'll have a significantly better IA.